Bismillahirrahmanirrahim, and welcome back to this next video, and this is the 13th video of the chapter 2 of Immunology. From the last couple of videos we are talking about the presence of the different type of the granules in the neutrophils, and I've told you that there can be primary, which are also known as the Azurophilic granules in the neutrophils. The second type of the granules that are present in the neutrophils, they are known as the specific or the secondary granules, and we talked about them in detail, that which kind of the antimicrobial components are present in the primary and in the secondary granules. In this particular video, I want to focus on the another type of the granules which are present in the neutrophils, and they are known as the tertiary or the gelatinase granules. These tertiary or the gelatinase granules, they are smaller granules when you compare them with the primary and the secondary granules, and they are fewer in number in comparison to the primary and the secondary granules. So these are two of the important distinguishing characters of the tertiary or the gelatinase granules, that they are smaller and they are fewer in number when you compare them to the primary and the secondary granules. Now these tertiary granules, they contain a special class of the proteases, a special class of the enzymes which are known as the Matrix Metalloproteinases. And these Matrix Metalloproteinases, they are responsible for the tissue remodeling by degrading the extracellular matrix proteins, which includes the collagen, the elastin, the gelatin, the matrix glycoproteins, and the proteoglycans. Why we call them as the metalloproteinases? Because when you look at them, for their function, you need the presence of a metal ion. For example, a zinc ion. So as they need a metal ion for their activity, therefore we call them as the metalloproteinases. Why we call them is the matrix metalloproteinases? Because these are the proteases which acts on the proteins which are present in the extracellular matrix. Therefore we call them is the matrix metalloproteinases. Now the presence of the tertiary granules that actually distinguishes the mature neutrophils from the immature forms in the bone marrow. Now the mature neutrophils, they typically contain these tertiary granules, while the immature form, they lack the tertiary or the gelatinase granule. So that can be one of the way to differentiate between the mature and the immature neutrophils. If you look at the matrix metalloproteinases which are present in these tertiary or these gelatinase granules, the most common matrix metalloproteinase found in these tertiary granule is the matrix metalloproteinase 9. For short, you can call them as the MMP9. This is the matrix metalloproteinase 9. It is also known as the gelatinase B, but that doesn't mean that these are the only matrix metalloproteinases present in the tertiary granules. There can be other as well. For example, there can be the matrix metalloproteinase 8. There can be matrix metalloproteinase 25, but the most common one and the most extensively studied of these matrix metalloproteinases is the MMP9 or the gelatinase B. Now, what is the mechanism of action of this MMP9? Because that is most extensively studied, I'm going to focus on this one. Now, the mechanism of action of the MMP9 that involves the degradation and the remodeling of the extracellular matrix proteins. Now, this ECM, this is actually the complex network of the proteins and the carbohydrates. Just giving you some of the examples like the collagen, the glycoproteins, the proteoglycans. So that this ECM, the extracellular matrix, is a complex network of the proteins and carbohydrates, and their function is to provide structural support to the tissue. Now, what this MMP do is that when it is going for the degradation or the remodeling of the ECM, so the first thing it do is it is going to recognize its substrate. And the MMP recognize and binds to specific proteins in the ECM, and the specific protein that the MMP9 degrade, that includes the collagen, the gelatin, the elastin, and the fibronectin. These are some of the important proteins that are degraded by the MMP9. Now these proteins, as I've told you, these collagen, the gelatin, the elastin, the fibronectin, they are the extracellular matrix protein. That means that these proteins, they form the structural framework of tissues, and they are very critical for the integrity and the function. Now, what the MMP9 do is, once it recognizes its substrate like the collagen, gelatin, once bound, the MMP exhibits its proteolytic activity, thereby cleaving the peptide bonds within the protein substrate. So first it recognizes its substrate like the collagen, gelatin, then it is going to exert its proteolytic activity to cleave the peptide bond within these protein substrates, and when the proteolytic activity has been completed, that would lead to the degradation of these proteins. Now the MMP that has a preference for specific amino acid sequences, particularly those rich in the protein, the glycine, and the hydrophobic residues. And these protein, glycine, and these hydrophobic residues, they are very common in collagens like the collagen type 4, 5, 7, and 10, and they are also very common in the gelatin protein. Now, by cleaving the extracellular matrix proteins, the MMP break down the structural integrity of the ECM, leading to tissue remodeling. Now, the question is why we need the tissue remodeling, why the MMP is going for the MMP9 is going for the degradation of these extracellular matrix proteins. By degrading the extracellular matrix component, the MMP actually helps create path for the immune cells like the neutrophils, the macrophages, the fibroblast, and the endothelial cells. It actually creates paths for the migration of these immune cells to the site of infection or injury. And when these immune cells that reach to the site of the injury of infection, the injury or infection, that can actually help clearing the infection in that particular area. Now, the MMP9, beside remodeling or degrading extracellular matrix component proteins, they are also responsible for modulating the activity of the cytokines and the chemokines. And the MMP that can actually cleave and activate, sometimes it can go for the inactivation of the cytokines and chemokines, and once they are activated or they are deactivated, this means that the MMP9 can actually modulate the inflammatory responses. Let me give you an example of this, how this can go for the activation of the cytokines. For example, the MMP can process and it can activate the pro-inflammatory cytokines like the interleukin 1 beta, the tumor necrosis factor, enhancing their biological activity. So when their activity is needed, the MMP9 is actually going for the activation of the interleukin 1 beta and the tumor necrosis factor alpha. Now, this MMP9 is also responsible for regulating the leukocyte trafficking. MMP9, it also facilitates the migration of various leukocyte subsets, including the monocytes, the macrophages, the T cells, the dendritic cells, by modifying the ECM and cleaving the adhesion molecules. So if you look at the baseline, what the MMP9 or these Matrix Metalloproteinases, they are doing, they are cleaving the ECM proteins, they are clearing the path for the entry of the immune cells like the neutrophils, in this particular case like the T cells or the dendritic cells, to the site of infection of injury, thereby performing their immune function. So that is the key. You're degrading the ECM proteins to clear the path for the immune cells to reach to the site of infection or injury. Now, this activity is very crucial for the immune cell, you can say, trafficking to the lymphoid organs or to the inflammatory sites. So if the ECM, extracellular matrix, that remains intact, these immune cells will not be able to reach to the site of infection of injury, but with the help of the MMP, you can actually clear path for these immune cells to reach to the site of infections or to the, say for example, lymphoid organs. Now, this is a very important phenomena, the neutrophil extracellular tract formation. I'll have a detailed video on that, and my next video will be focusing on this neutrophil extracellular tripher formation. But generally, if I tell you, the neutrophil, they release web-like structures which are called is the neutrophil extracellular traps, and these traps are very important to trap and kill the pathogens. How that work, I'll have a detailed discussion on that in my next video. Why the function of the MMP9, that is very much important, because the dysregulated MMP9 activity that has been implicated in numerous inflammatory and autoimmune diseases, and if the dysregulation of the MMP9 is involved in the inflammatory and in the autoimmune diseases, that means that its regulation, its normal activity is very important in the immune functions and in the disease pathology. So that means you need the MMP9 for a normal immune system. If you like the video, please subscribe to my channel, hit the notification bell. If you have any question, you can ask me in the comment section, and do share it with your friend. I'll see you in the next video.